Good afternoon, everyone, to our colleagues and our guests for this session. So I'm Dr. Francisco M. Heralde, a professor from the Department of Biochemistry and Molecular Biology, College of Medicine, UP Manila. This afternoon, I'm going to give you a lecture on the evolution of diagnostic testing in COVID-19 pandemic. So, as we know, we are now in a pandemic, and we've been around since the last 2 years. So this all started in December 30, 2019. As we know, it all started with a report in Wuhan about a case of flu and pneumonia, severe pneumonia, and followed by mortalities. And from then onwards, in 2019, this spread to other countries in Japan, Korea, and we had our case in the Philippines in February 2, which is a first death reported outside of China. And this has spread to other countries, Brazil, Europe, South Africa, such that by September 25, 2020, it has spread globally, resulting to around like 980,000 plus deaths. In the Philippines, as of July 2, we have a reported confirmed cases of like 1.4 million and deaths of around 24,800. And we have been informed all along that we are already in the second wave and managed to like maintain our daily cases. So this is now like under the new normal. And of course, we have to know what is the cause of this pandemic. So as we know, this is caused by the SARS-CoV-2 virus, and this virus is an RNA virus with around 28,000 base genome. It's enveloped, and it belongs to the coronavirus genera. And there are 4 under this, mostly like the alpha, beta coronavirus, gamma, and delta. And usually the alpha and beta are found in mammals, and SARS-CoV-2 belong to the beta coronavirus. It is said that the natural reservoir of SARS-CoV-2 are the bats, and there is an intermediate host which is said to be pangolins. No? And as you can see here in the right panel, this is the structure of the schematic diagram of the virus with the RNA inside the envelope, and there are spike proteins protruding outside this structure. And if you look at the genome, there are like set number of genes. You're talking of like ORF1A, ORF1B, or open reading frame. So it means, and then you have the spike gene, the envelope gene, the M gene, so and so on. And within the spike, you have the RBD, or the receptor binding region. And usually these are the targets for the molecular characterization of this virus. Okay. So apparently what's so special about this virus is that it possesses the ability to bind to the ACE2 receptor, a human ACE2 receptor, and that's why it became highly infectious. Okay, so now there are several testing methods used for this virus, and at least here, what is known or considered as a gold standard is the nucleic acid amplification test, or the molecular methods to detect the virus. So this is the preferred method because it's relatively sensitive enough. Uh, Although there are numerous molecular tests, so and usually this test gained emergency use approval from the FDA, and there are target genes for this virus, the nucleocapsid ORF1ab envelope, or the RDP, or RNA-dependent polymerases, and the sample is usually obtained from the upper or lower respiratory tract, the NPS or OPS. However, there are many challenges associated with the collection device as well as the reagent supply at the start. And the other way that this virus is tested is through antigen detection, and this use viral proteins, recombinant proteins, and then there are antibodies generated against that and used to target those proteins produced by the virus detected from the nasal or nasopharyngeal swab. And the other, while this one is essentially a quick kind of point of care test and uses the lateral flow immunofluorescent assay platform. This is reported to have a relatively acceptable sensitivity of around 80% and 100% specificity. And although several independent evaluations of accuracy still needed. Now, here in the schematic illustration, as you can see, there are two sides or two types of tests. We have the nucleic acid amplification test using our swab in a placed in our tubes, with swab with the transport medium, viral transport medium, and then you have your DNA or RNA of the virus, and then detected through the lateral flow or to the PCR. And then, of course, you have serology to detect the antibody present, and also there is a lateral flow test using pinprick to detect the antibody IgG, IgM. And this is correlated with the clinical assessment of like tomography, chest X-ray, etc. So that is how the SARS-CoV-2 is being diagnosed from the patient and in the population. Now, of course, it has to be understood that there is a timing in all these tests, like both in the PCR as well as in the antibody response to SARS-CoV, because there is a certain pattern in which the body responds to the virus. So it's a new virus, so there are no pre-existing antibodies or immunity in our body, and we're still learning about our immune system to this immune response to the SARS-CoV-2. And it is said that many develop antibodies 1 to 2 weeks after the symptoms, and greater than, and this apparently is due to the delay in the seroconversion. However, the antibodies do not play a routine role in the diagnosis. And also, 95% of the patients are antibody positive after 2 weeks. Some patients may not seroconvert, and so the immunostatus is critical, as well as this is assay dependent and the severity of the illness. And it is said that the viral RNA would persist up to 6 weeks, and the symptoms onset occurs after a week, and the IgM declines 5 to 7 weeks post-onset, while the IgG remains positive for greater than 10 weeks. Okay, so here in table 1, we summarize the different PCR-based, RT-PCR-based kits developed in the different institutions. And as you can see, the kits are not the same. They have different targets. And usually the most target is ORF1AB and N. So you have ORF-AB codes for the polymerase, and the N is the nucleocapsid protein. And one of the assays where I'm very familiar of is the BGI, because this is the one we use in our WHO study, and apparently it targets the ORF1 gene and has a sensitivity, a limit of detection of 1 to 2 copies per reaction. Okay. Now, there are other nucleic acid amplification tests aside from RT-PCR. The loop, isothermal PCR. In other words, you don't need a PCR machine. And so like the enzyme would make several copies, and then these are detected through the lateral flow platform. And also there is a CRISPR-Cas based kind of assay. So this capitalizes on certain property of the virus, or rather sequences in the virus that is able to detect repetitive sequences, the CRISPR or the So and then this is capitalized for the detection of the virus using either plate reader or lateral flow visualization. And then finally, the other one uses the gold nanoparticles. And then, so apparently these gold nanoparticles are conjugated with oligonucleotides, and this one would hybridize with the viral RNA and pull them down and develop some kind of a visualizable chemical reaction. And of course, the other type of platform involves the following. So you can see we're more familiar with these terms like ELISA, enzyme-linked immunosorbent assay. So there is an antibody-based kind of test, and with an enzyme reporter molecule used to detect the presence of the antigen or the antibody. And then we have the lateral flow immunoassay, and then also the electrochemical sensing, as well as microarrays. So, and then in the ELISA, it can be either in the format wherein antibodies are detected, the IgG or the IgM, or it could be in the format wherein the antigens are detected in a sandwich platform. So the plate is pre-coated with the antigen-specific antibody, and then once the antigens are captured, there's a secondary antibody that would conjugated to a reporter molecule to detect it. So essentially that. Now, of course, some in table 2, you can see some of the available detection tests based on the immunoassays. And you can see that several of them uses the lateral flow platform, and they have different antigenic targets or antibody targets as well, and uses the technologies we have previously discussed. And here in this table, this is the more sophisticated form involving automated commercial antibody detection kits. So this now uses integrated machine and then high throughput systems, such that this would increase the turnaround time and batch run of samples of like nasopharyngeal samples. So the ones that we are very familiar of are the like one tie ELISA platform, and the other is the Biorad Platelia platform, because these are the ones used in our study. So you can see this use serum plasma and use sandwich ELISA, but they have different targets, like one tie targets RBD and Platelia targets the nucleocapsid protein. So what are the factors driving the evolution of COVID-19 diagnostics? So I have listed at least 3 factors here. So I lumped them up, like in the first factor, this includes convenience, cost, and time. And as you know, the most the gold standard is the NPS OPS specimen, but you know that this is too invasive and there are other safety issues, and because the more frequent the sample is collected from the NPS OPS, so there's a danger that it could damage the nasal tissue, and so that's why other strategies have to be ventured or sampled. The other thing is that RT-PCR is a bit costly, pricey, while the lateral flow is much cheaper. And also the requirement for a shorter turnaround time. Now, of course, the other factor that is happening now is the occurrence of the mutations of the virus, and leading to the establishment of the other variants. And as we know, we have more than 6 variants. Now we have alpha, beta, gamma, delta, P, and then P.3 and the delta plus. So, these variants are more aggressive and modifying the rate, the severity, response to the vaccine, as well as the pathological pattern, no, of the disease. And then the other factor is the vaccination and the asymptomatics, because as we know that different vaccines results to different immunity status, and a possible vaccine escape variants, like what happened in South Africa, or in the AstraZeneca, they decided to stop because the beta variant showed some resistance to those that were vaccinated. And the other thing are the asymptomatics, which can possibly be some kind of like a breeding ground of the virus, because they don't know that the virus is in them, and yet it remains there. No, okay. Now, of course, among these patterns in evolution of the COVID-19 diagnostics, so there, in response, as you can see, there are emergence of variations in sample testing, like the emergence of saliva tests. So there are data that says that instead of doing the conventional NPS OPS, so we use the saliva test, so it's not as invasive, and other swab samples aside from the NPS OPS, like the rectal or anal. So this has been an issue, for example, in China, where in Japanese diplomats had to undergo rectal anal, and this had some like some kind of ethical question, especially in port of entry in airports, no? So cervical, placental, breast milk, cord, blood, amniotic fluid. So these are the non-conventional samples, which also we have optimized, and we've been testing in our study. And then the emergence of artificial intelligence-based tests. No? So no need just for example, a saliva or sweat or a breath, they use dogs, for example, or like or other machines, no, like to calculate for the like certain markers in the pattern of the saliva, and the artificial intelligence data is processed by the computer to detect the presence of the virus. And then, as we said, as more variants come in, so instead of just using the PCR now, the need for sequencing, so like the Genome Center is doing the next generation sequencing of the variant. But of course, this is more expensive and there is a limit to the number of tests that they can do to detect the variants. Now the option is direct PCR and sequencing of the target genes, but this has been set up and only a small number of laboratories are engaged into this. No? And then, of course, the vaccination, we know that we have more than 6 types of vaccines that is being rolled out. And although we have already more than 10 million of Filipinos vaccinated, but the different vaccines have different antigens in their composition, and this results to different efficacy and neutralizing antibody. And this would modify the kind of immune response generated to the vaccinated individual. Of course, for the asymptomatics, there is natural immunity and delayed immune response, no? So which also could modify the pathology or the pathologic pattern of the virus. No? So, and this all impact on serology testing. That's why in our study, we're using different types, 2 types at least of ELISA testing. One that for the unvaccinated anti-S, which is 1 tie, and then the other one is for the anti-N, which is Patelia. Now, so all along, all these diagnostics that came out, in for testing during the pandemic has been established and has to undergo the evaluation system of RITM. So apparently, RITM is keeping track and mandated to assess the efficacy of the different tests. So starting with SARS-CoV-2 plate-based PCR, and then SARS-CoV-2 cartridge-based PCR, and then we have the viral RNA nucleic acid extraction kit, the SARS-CoV-2 equipment-based antibody test, SARS-CoV-2 rapid antigen test, SARS-CoV-2 rapid antibody tests, as well as the viral transport media. And as you can see, this is available in their website. And so open to the public, so you can look at the list of the kits that were approved starting from the earliest time of the pandemic up to the present time. And they have a standard procedure for evaluating, for example, in the qPCR. So they have a minimum of 30 positives and 30 negatives, and also like there is a standard extraction procedure that is associated with this qPCR-based platform. And you can see they have also in the website the guidance of WHO, wherein the clinical sensitivity is greater than 95 for these kits, as well as the specificity should be greater than 99. And this is the requirement also set by DOH and the Health Technology Assessment Council. Okay, so here you can see the list. So starting with April 1, 2020 and onwards, so different kits have been evaluated. And of course, during the early stages of the pandemic, what was so controversial then was the Manila Health Tech, or from the UP Manila Gene Amplify kit, which actually delayed the Marikina rollout of the diagnostic testing because the initial result in terms of sensitivity was low. So it failed the standard set by WHO. So that's why they had to resubmit the kit or to produce the version 2. Now, of course, here you can see the BGI real-time PCR, which was used in our WHO funded study. It passed the sensitivity and specificity requirements. And the one that we're familiar, also I'm more familiar, I work with Lung Center as a visiting consultant. So Sansure was the one that's also popularly used there because they come with the machine and high throughput testing of specimens and so on. And so here you can see the SARS-CoV cartridge-based PCR, and you can see that starting May 2020 to March 2021, only 6 registered for this. So, and you can see the first one that registered in May was the CFID, which is the gene expert. And we know that the gene expert is this is unlike the plate-based. So this one is there's a cartridge, and like depending on the machine, you can run like there's a 4 cartridge or 6 cartridge machine, gene expert, which is actually the platform, the machine has been used for quite some time for the TB program, and it was also easily converted to SARS-CoV testing. And of course, we have all the other platforms like CoVAS, which came out later. And then for the nucleic acid extraction kit, you see it's a Kiagen viral RNA mini kit is being used. And it was registered early in April and seemed to have passed in all the criteria set. And yeah, and as well as all the others. And in our case, like in our study, we use the MGI nucleic acid extraction kit. And so far, it has undergone like passing rate in at least 2 of the criteria as of May 2020, and so on. And then there is also the antibody equipment test. So far, this was only 2 registered for this in 2020 November, and also for the other kits like so this is antigen kit. So several have registered, and either this use the dry fluorescence immunoassay or the fluorescence immunoassay type, or the test the strip type or the lateral flow assay type. And also you have the rapid antibody test. So we're more familiar of this one because this is a point of care, and you can see that there's really a battery of tests being done, whether it can detect at the early, mid, or later stage of the infection, and then whether it can detect IgG or IgM, and what is the specificity. And this testing has been like started like in April and then 2020. And you can see, for example, that one Thai SARS-CoV antibody is one of those that have passed the rigorous testing. And in our study, for example, we use Celex, and so far, like Celex is a bit well, at least it was acceptable, around 89 sensitivity and specificity to 9. And then also now what is more commonly used is the Clown gene, and so far it has a acceptable specificity and sensitivity both for IgG and IgM, and this was registered in December 2020. Now, so this is the summary of the evaluated diagnostic test in RITM. So you can see that so for the different categories, the ones that are, there are several players in this diagnostic space. Usually a SARS-CoV plate-based PCR, you have 34. And then the rapid antibody tests, you have 39. And the registering has continues, no? Starting with April 2020 and onwards. And new and improved versions are continue to be registered and evaluated by RITM. So as more tests come, and they have to address the three issues that we have raised previously: convenience, the time, and then also the variants that are emerging, and as well as the impact of the vaccination. So with that, I'd like to end my lecture. Thank you very much for listening.